Hello students, I hope you are doing fine. So today I'm here and I will be talking about the primer design. So the word primer is very common to all of you, and I believe that you remember this because we have discussed primer or used the word primer several times in different types of PCRs as well as during the various sequencing reactions. So a primer is a short or small single-stranded nucleic acid sequence, which is generally utilized for the synthesis of DNA, or it is utilized for the replication of the DNA. The primers are either used in the living organisms or they are used in artificial medium such as during the PCR reactions or sequencing reactions. Now, during the various biological processes or during the process of DNA replication inside the living organisms, primer is required for the initiation of the process of DNA synthesis. Similarly, in case of artificial environment also, primer is required for the initiation of the DNA synthesis. Now, why it is required for that purpose is because we know that the DNA replication or synthesis is brought about with the help of an enzyme called as DNA polymerase. And very importantly, this DNA polymerase cannot start the process of DNA synthesis as such. That means it cannot start the process of DNA synthesis by itself. It needs an existing strand or an existing template where it can attach to and bring about the addition of the nucleotides. Okay, so therefore we need this short sequence called as the forward primer or the reverse primer during the artificial environment also, or during the inside the living organism also during the process of DNA synthesis. And to this existing stand, the DNA polymerase can bind and bring about and carry out the addition of the nucleotides at the 3' end, both at this location as well as at this locations. So that is why the primers are very important. So I think you're familiar with this structure or this picture that is shown over here in this slide, wherein it is showing that how the DNA replication is being carried out and the replication fork is formed during the process of DNA replication in the living organisms. And if you see closely, then there is something over here, and this is called as RNA primer. Okay, so what is this RNA primer? RNA primer is the primer or is the single nucleotide sequence which is helping in the process of DNA replication inside the living organism. Okay, so RNA primers, they are used in living organisms in the initiation of synthesis of a DNA strands, and primases add a complementary RNA primer to the reading template on both the leading and the lagging strands. And I hope I do not have to talk about what is a leading strand and what is a lagging strand. You are very well familiar of this. Okay, now synthetic primers are different from the natural or the biological primers in the sense that they are not synthesized or they are not formed inside the living cells, rather they are synthesized outside by the process of chemical reactions. Okay, now this property by which or the chemical reaction by which we are able to actually synthesize the primers has been proved to be a very invaluable tool, specifically in the field of molecular biology. And as a result, we are able to carry out different molecular biology experiments, that is, PCR reactions, RT-PCR reactions, different types of PCRs that we have already discussed. And we have also been able to carry out the sequencing using the old generation or the first generation, second generation to the third generation sequencing, as well as the fourth generation sequencing techniques. All of them, they utilize primers. So the primers are very important, and therefore synthetic primers are of great use to the field of molecular biology. So when we talk about the PCR primers or the primers, what is the general requirement for a primer? See, the requirement of a primer may vary depending upon the type of application of the molecular biology, but by and large, the basic important characteristics of the primers remains the same, and they are: so for an efficient and reliable PCR, and if we want to achieve a specific amplification of a particular product, then this can be done by using the DNA primers. And very importantly, some of the steps must be kept in mind while designing the primers. The first thing is that it is not necessary, remember, to design the primer every time. Sometimes what happens is that whatever study or whatever research you are carrying out, generally people have already done research in that field, and some of the primers, they are already available in the public domain. So before you actually start making your own primer sequences, I'm not saying you are doing a chemical reaction. So you have to be, for a primer design, that means when I say primer design, it basically means that you are finalizing the sequence of the nucleotide that you need for to carry out either the PCR reaction or sequencing reaction, etc. Okay, so you are finalizing that sequence. But before you finalize that sequence, kindly search the literature, kindly search the database. If it is already available, it might be that you might not even need to design your own primer. Okay, okay, let us assume a situation that you do not have a sequence in the database. Okay, in that case, to choose, be sure what is your target sequence, what you want to amplify. Do you want to amplify a RNA sequence? Do you want to amplify a mitochondrial gene? Do you want to amplify a particular gene, for example, DNA polymerase gene? So depending upon that, you have to choose your target sequence, and then using that sequence you have to design your primers in such a way that the primers can specifically bind to that sequence only and does not bind to any other part of the genome. Okay, so this step is very important. You do not, the bottom line is, जो मैं कहना चाहती हूँ कि जरूरी नहीं है आपको हमेशा प्राइमर हमेशा डिजाइन करने पड़ेंगे अपनी स्टडी के लिए. लिटरेचर में कई बार ऑलरेडी अवेलेबल होते हैं प्राइमर्स, सो गोइंग थ्रू द लिटरेचर इज वेरी वेरी important. Okay, now talking about the characteristics of primers. So generally for a conventional PCR reaction, हमारा amplicon, it generally ranges from 200 to 800 base pair, or we can say that from 200 to 1000 base pairs. But आज के time में ऐसे भी PCR reactions हो गए हैं which can amplify a larger fragment also, but this is on an average. However, in case of real time, the range is between 75 to 200 base pairs. 75 से नीचे नहीं जाना है. We don't go below 75 because the same band, we want to have a band which is slightly above the primer dimer bands. Okay, so that is the reason. Now, what are the characteristics of a good primer? There are several characteristics of a good primer, and we will be discussing about them one by one. So generally, a good primer must have a GC content between 40 to 60%. मतलब उसमें if you count all the G's and all the C's in the sequence of a primer, then they should not be more than 40 to 60%. The reason is because you know that G as well as C, they form triple hydrogen bond. And you know that during a PCR reaction, there is first annealing of the primer, and then during the cyclic process, there is again denaturation of the DNA, and then there is renaturation. So the point is that if the primer will bind very strongly to the DNA sequence, it will be difficult to break the bond. Okay. And another important point is that the melting temperature of the primers, it must range between 50 to 65 degrees Celsius. I'll talk about it later. Okay. So why we are so this I have already discussed that the GC content should be 40 to 60%. So if the GC content is very high का मतलब है if the primer is having very high GC content, then it will look for the sites and bind to the sites which will have a and the bonds formed there will be very stable. Primer गिरेगा नहीं वहां से. Okay.